Hi everyone, welcome to SPARQ-ed's virtual lessons on working with DNA. This first module is an introduction to DNA and the tools of biotechnology. There are two presentations in this first module, Presentation A, What is DNA? And Presentation B, Biotechnology, Recombinant DNA and Cloning. Ideally, as you work through this presentation, Presentation A, you should also have Booklet A on hand to work through. You can write your notes and answers directly into a hard copy of Booklet A, or if you don't have Booklet A printed, you can write your notes and answers into your own notebook. The learning intention for this presentation is to understand that DNA is a double-stranded molecule that contains genes. By the end of the presentation, you'll be able to define the term gene. You'll recognise that the purpose of gene expression is to synthesize a functional gene product. And you'll be able to identify differences and similarities between DNA in eukaryotes versus prokaryotes. Now, DNA is in every living thing as well as in some non-living things such as viruses. So it's really important to know about. Read through these 5 statements about DNA and decide whether you think they are true or false. How did you go? Let's go through the answers. DNA is only found in animals. This is false. DNA is found in all living things. DNA can replicate to make copies of itself. Yes, this is true. DNA can be cut and pasted. Yes, this is true. This is what you'll get to learn about in Presentation B, about recombinant DNA. Many viruses contain DNA. Yes, this is true. Even though viruses technically aren't living things, many contain DNA. Those that don't have DNA contain another nucleic acid called RNA. Finally, all DNA has the same overall structure. This is true, and it can be seen in this diagram. DNA is a double helix, it's double-stranded. The helix is the twisting shape. The backbone, the red and blue lines on the side, are made of phosphate and sugar. And the letters in the middle are nucleotide bases. A always matches with T, and C always matches with G. Okay, so let's have a look a bit more closely at what DNA actually is. DNA is a physical molecule. It's a substance that contains a code to make proteins. Proteins are the molecules of life. They catalyze reactions and make structures in your cells. You are pretty much made of protein. You can think of proteins as the building blocks and workers of your cells. DNA and proteins are found in the cells of all living things. So here we have an imaginary cell. It must be a bacterial cell because there's no nucleus. There's a molecule of DNA, double-stranded DNA. And the DNA has some sections called genes. Now, a gene is a section of DNA that codes to make a protein. The gene can be transcribed to make mRNA, messenger RNA, and then the messenger RNA is translated to make protein. If you want to know a bit more about these processes, please follow our links to the University of Utah websites, which have some great information about transcription and translation. Now, it's commonly said that if we were to stretch out all the DNA in just one cell of our body, it would reach to 1 meter long. So with so many cells in our body, that means we must have a lot of DNA. How on earth do we fit all that DNA into our cells? Well, DNA is tightly wound into these packages called chromosomes. In this image, we've got a eukaryotic cell on the right. In eukaryotes, DNA belongs in the nucleus, and the chromosomes are structured around these proteins called histone proteins. In prokaryotic cells, such as bacteria, there is no nucleus. The DNA is wound up, but just used loosely in the cytoplasm. Have a go completing page 3 of booklet A before moving on. As we go through the next few slides of information, take some notes in your booklet on page 4 or in your notebook. You don't need to copy things down, just take some dot points and maybe draw a diagram. Here, we can see that bacteria, as mentioned before, have DNA. And as we learnt before, the DNA is organised into a chromosome, even though that chromosome is quite different to the chromosomes we have in eukaryotic cells. Now remember that bacteria are prokaryotes, so they don't have a nucleus. In this diagram on the left, the purple stuff there is bacterial DNA. On the right, you can see an electron micrograph of a bacterial cell. So that's an image taken from an electron microscope. Now, in addition to their regular chromosome, bacterial cells also often contain a small circular piece of DNA called a plasmid. Bacterial cells can share plasmids between each other, and therefore they can share the genes that are on those plasmids. These days, plasmids have been adapted for biotechnology, and we can buy specific plasmids from commercial stores online. We use these plasmids as DNA vectors. Another place you would have heard the word vector before in biology is when studying diseases. In disease biology, a vector transports a pathogen from one host to another host. For example, mosquitoes are the vectors for transporting the malaria-causing parasite from host to host. In this case, however, a vector is a bacterial plasmid that transports a gene from one organism to another. This green diagram here represents a bacterial cell. In addition to its regular chromosome, in red, it also contains some plasmid molecules. How many plasmids does this bacterial cell contain? Also, what do you notice about how the DNA molecules are drawn? Why are they all drawn with 2 lines? Have a think about that and take some notes on page 4. Here you can read through 8 statements about DNA. For each statement, you need to identify whether they apply to prokaryotes, such as bacteria, or eukaryotes, such as animals, or both. Well done on getting through the first presentation. And thank you for listening. If you do have any questions, please feel free to email us, and we hope to see you one day at SPARQ-ed.